speaker is Caroline Pierce from the Department of Soil, Water, and Climate. Thanks for being here, Caroline. Please go ahead.
Yes, you can see my screen?
Yes.
Okay. So hello, and thanks for having me. I'm Caroline Pierce, and I'm a fourth-year PhD student in Land and Atmospheric Sciences. Today I'll be talking to you about sulfur speciation in peatlands and how it might affect the neurotoxic form of mercury called methylmercury. So to start off with some background, mercury is a widespread and long-term pollutant. Mercury is emitted to the atmosphere through natural sources like volcanic eruptions and forest fires. But since the Industrial Revolution, atmospheric mercury has increased 3 to 5 times because of the burning of fossil fuels and other industrial uses. This atmospheric mercury can cycle the globe for centuries to millennia. Even though since the 1970s, we've drastically reduced the amount of mercury we emit, that previously excess emitted mercury is still in the atmosphere. This mercury in the atmosphere is then deposited into terrestrial and aquatic systems where mercury can be transformed into methylmercury. Methylmercury is toxic at environmentally occurring levels and can have human health impacts. Within peatlands, mercury is methylated as a byproduct of certain anaerobic microorganisms, sulfate-reducing bacteria, iron-reducing bacteria, and methanogens. Keep in mind that as mercury is methylated, it is simultaneously demethylated. So the balance of methylation to demethylation leads to a net accumulation of methylmercury. Once methylmercury is produced, it can be slowly exported to surface waters where it biomagnifies in the food web to top predatory fish. Humans and wildlife who then eat these fish can suffer from neurological disorders, especially children and pregnant women. Moving on to peatlands. Boreal peatlands are organic soils that are permanently or semi-permanently saturated in cold climates. So peatlands are a sink for mercury because of the carbon and sulfur that is present in peat, and these three elements have a strong affinity for each other. Peatlands are also ideal environments for the production of methylmercury. It's anaerobic, there's mercury, and there's microorganisms that can produce methylmercury. Sulfate-reducing bacteria are considered to be the main contributors to methylmercury, so that's why this research topic is focusing on sulfur. Additionally, another aspect of peat that's relevant to this study is the topography of peat. You can see a picture of this on the bottom right-hand corner. These are called hummocks and hollows, and hummocks are mounded areas that alternate with wet hollows, and these two topographies have different conditions based on their relative positions to the water table. So our objective for the study was to characterize the interactions of sulfur and mercury in a boreal peatland. Past studies have demonstrated that an increase in sulfate will lead to an increase in methylmercury production. What we don't know is how this sulfur is transformed in the peat as a result of the mercury methylation process. The main findings of this research are that we've identified potential reactants and products in the methylation process. Additionally, we found evidence that some sulfur species may actually affect the bioavailability of mercury, resulting in inhibited methylmercury production. To touch briefly on methods, we examined the size, composition, and spatial correlation of sulfur and mercury pools in peatland soil profiles. These samples are from the Marcel Experimental Forest, which is near Grand Rapids, Minnesota. Seen on the top left are surface depths of the peat cores. We took peat cores to a depth of 200 centimeters, and this picture represents the top 30 centimeters of the peat core. On the right, you can see me purging vials of peat with nitrogen to remove all oxygen from the sample containers. And a really cool part of this research is how we determined the sulfur speciation. For sulfur speciation, we used sulfur 1s absorption, X-ray absorption near-edge spectroscopy, also known as XANES. Essentially, we bombarded the sample with X-rays at different energies And this produces a spectrum for the sample that represents the sulfur species present. You can see this on the bottom in panels A and B. To figure out what these sulfur species are, we have a reference library of pure sulfur compounds, and you can see that in panel C. Through linear combination fitting, we determine what combination and proportion of the references best fit the sample spectrum. We also determined total mercury and methyl mercury concentrations through standard EPA methods, and potential methylation and demethylation rates were determined by enriching the peat samples with stable mercury isotopes. So our main findings were that organic disulfide, which is a reduced sulfur species, is positively correlated with methyl mercury. This means that as organic disulfide increases, so does methyl mercury. This may indicate that they are co-products of the mercury methylation process via sulfate reduction. Organic monosulfides, which are also a reduced sulfur species, are negatively correlated with methyl mercury. This result indicates that these species may decrease the bioavailability of mercury 2+ to be methylated. Finally, ester sulfate is positively correlated with methylation rate constants, which indicates ester sulfate may be a substrate for mercury methylation. I'll go into more detail on all of these, but let's start with the organic disulfide finding. On these figures to the right, you can see the depth profiles of methyl mercury and the different sulfur species. These are separated by hollows and hummocks, which I mentioned earlier. And in the sulfur graph, the relevant line is the purple line, which represents organic disulfide. I want to point out that the maxima for methyl mercury and organic disulfide occur at the same depths. And like I said, the disulfide is the purple line. So we also used Spearman correlation coefficients. The way correlation coefficients work is they range from -1 to +1. A value of 0 means that there's no relationship between the two variables. A high positive value indicates a strong positive correlation, and a negative value indicates a negative correlation. So in this table, I've only listed variables that had significant p-values, and the correlation coefficients are labeled as rho, and anything that's positive is in black, and anything that's negative is in red. So these are all the variables we compared to methyl mercury. And you can see for hummocks, there was a significant positive correlation between organic disulfide and methyl mercury. However, this result is not seen in the hollows. There are a couple reasons that this could be. So hummocks and hollows are different in terms of their distance between the peat surface and the water table, their geochemical environments, and the density of the peat. All of this results in hummocks being more oxic than hollows. And like I mentioned before, to produce methyl mercury, it has to be anaerobic. So these results may be accurate, that it only is a trend in hummocks, but I think it has more to do with our sampling plan. If you look at the depth profiles, you can see that there are more sampling points in the peak area for the hummock, which makes it more resolved. It's possible that if we had included more sampling points within the peak area of the hollow, we might have gotten that same trend with organic disulfide. So to summarize, organic disulfide is a possible end product of microbial sulfate reduction processes in peatlands and may be a co-product along with methyl mercury. Moving on to the second finding, organic monosulfides, which are composed of thiols, monosulfides, and thiophenes, are negatively correlated with methyl mercury across both hollows and hummocks. This was a surprising result because we were expecting all reduced sulfur species to be positively correlated. This is because methyl mercury is produced as a byproduct of sulfate reduction, so we thought all reduced sulfur species would be produced along with it. If you look at the depth profiles, organic monosulfide is in yellow. Organic monosulfides have maxima in the lower depths where methylmercury concentration is low. So a subsection of organic monosulfides are thiols, and thiol functional groups are known to strongly bind to mercury 2+. For methylmercury, it's possible that this strong binding affinity between thiols in the peat and mercury 2+ actually causes a reduction in the bioavailability of that mercury to be methylated. So on to the rate data. The depths at which the greatest methylation rate constant, called Km, and that's in the top right corner. The greatest rate constant occurs in the range -10 centimeters to -20 centimeters below the surface. And this is in the same range that you would see a peak for methylmercury concentration. So Km is positively correlated to ester sulfate. This provides evidence for ester sulfate being the reactant in the methylation process via sulfate reduction. The ratio of the methylation rate to the demethylation rate, called K ratio, which is in the center, is positively correlated with ester sulfate, which matches this finding with Km. Adding further support to our previous finding about organic monosulfides, K ratio was also negatively correlated with the organic monosulfides group. So our findings point to the importance of organic sulfur species in the mercury methylation process as substrates and products, as well as potential inhibitors of bioavailability. In addition, for a peatland system with sub-micromolar pore water concentrations of sulfate, our findings indicate that the solid phase sulfur pools, which have a much larger concentration range, may be accessible to microbial activity and/or exchanging with the pore waters over short time scales. This information also gives us baseline information about this system, which will inform our future work, which involves how climate change will impact these systems. Are there any questions?
Thanks so much for that, Caroline. I appreciate it. It was a great presentation.
Thank you.
If you have any questions, please go ahead and unmute yourself or place them in the chat. I do have one question for you, Caroline. Is this work related to the spruce project as well?
Yes, so this is essentially a precursor to the spruce project. So this was, all of these samples were collected in 2012 prior to the initiation of spruce. So it serves essentially as like a time zero characterization. So these samples were taken from the location where the spruce project was built. And for those of you who don't know, the spruce project is a large-scale climate simulation experiment where there's different temperature and CO2 manipulations. So we're going to see over time how increased temperature and increased CO2 affect different aspects of peatlands.
Yes, thank you so much. And we do have one question on the chat for you, Caroline, from Jake Bailey. Would it be accurate to say that the monosulfides are mostly primary biological molecules, whereas the disulfides are the result of a diagenic reactions with sulfide?
I'm not sure I know the answer to that. From my understanding, so the way sulfate reduction works is it produces hydrogen sulfide, and that can be incorporated into the peat. So I'm not sure if organic monosulfides are the result of microbial activity or if it's a transformation that happens after it's already incorporated to the peat. So that would have to be a future study to look into that.
Mm-hmm. Thank you so much for that. Appreciate it.